It's visible.
That's
Yeah.
fine, right? Yeah. Okay. So I'll stop sharing now, and whenever we are ready, I can share it.
So we'll start at 9:30 sharp, sir. Umesh.
Okay.
Thank you.
So I'll put myself on mute and.
Okay, sir. Thank you.
ನೋಡಿದೀರಾ A bit of late, sir. The students has to yet to join.
No problem, no problem. No, I was just trying a different camera. But,
Nowadays, students are like that, sir.
They all late risers.
Yeah. So you are in contact with the Venkataraman, sir?
Oh yeah, yeah. We exchange messages quite frequently. I met him in person a couple of months ago in Florida.
Previous week, I talked to him.
Okay.
He's very much interested in, I think he may join now.
Oh really? Okay.
Yeah, he may join now.
So you have shared the link to.
Yeah, I have shared a link to him.
Okay.
He is very much interested in this, especially in this program. That too, Murali involving in this program. I asked him to, he also can join and give a guest lecture.
Yeah.
I don't know why he declined.
Uh,
Tomorrow, another Jay Kumar, sir, is also the CEO one good at US company.
okay.
Maybe Pepsi company is a CEO like that, sir.
Okay.
Vice president.
He's also
alumni.
okay.
He's 81 to 85 batch, sir.
Oh, okay. A couple of years after.
Yes. He's giving that one lecture.
Okay.
And one more, Swaminathan, sir. He's agriculture scientist at Texas.
Okay.
Alumni. Day of tomorrow, he is delivering one more lecture. 3 plenary sessions are there. Yourself, Jay Kumar and Swaminathan are delivering the special lecture.
Okay. Nice to be
Yes, sir. Group wise, we have made it, sir.
okay.
Group wise, the PhD people, students will deliver oral presentation.
Mm-hmm.
They are evaluated and
Oh.
they will be given the certificates. And like that for MSc also, sir.
Yeah.
They have made a poster presentation.
Okay.
For poster presentation also in the corridor where you are studying in the microbiology.
Yeah.
Black auditorium. I think you may remember they in that corridor we are displaying all the
Posters. Yeah. No, it's a nice place.
Yeah. So that is all classrooms are there. 1, 2, 3, 4 classrooms are there. After that, space is there. And there we are displaying the posters. They also evaluated. Like this 4 days program, sir.
Okay.
Uh,
And then there will be best award for best poster and all of
yes,
that.
sir. Yes.
Okay.
They are recognized with the certificates.
That's wonderful.
Yes, sir. That's it is been there since 2014, sir.
Okay. In our time, during our time, it used to be just in the department itself.
Yes.
Now this is good. They get also a little bigger exposure. They also
Yes.
get to hear from people from outside.
Yes.
Farmers. It's good exposure.
Yes, sir.
And for them also to present in front of a bigger group.
Yes.
Nice.
Only the students are good, sir. They're excellent. Only thing is exposure is required.
Yeah.
Good exposure. Dr. Ganeshaya started this program.
Okay.
You know Ganeshaya?
Yeah. Ganesha.
Ganesh.
In physiology.
Physiology, yeah.
No, not Somshekhar. Sure.
Dr. Uma Shankar.
Uma Shankar. Yeah, Uma Shankar. That's what I was. Yeah, they were all senior to us. They were all quite.
Yeah. Uma Shankar.
They must have retired now, right? They all.
Yes, sir. Yes. Is Dr. Bhanu Prakash, sir.
Okay.
Dr. Bhanu Prakash is a scientist in sericulture.
Okay.
As a professor there. Now he is working as our scientific officer in our directorate, post-graduation studies.
Okay.
Yes, sir.
Nice to meet you.
Such. Right now you are in Bangalore only, sir, or somewhere outside?
I'm in Bangalore.
Thank you.
I'm traveling tonight to US, so,
Okay.
but I'm mostly based in Bangalore.
Okay. Sir. So it's 9:30. We will start. Otherwise, the program, Sir Dean, sir, we will start, sir. Otherwise, the further program will get delayed. Mute ಆಗಿದ್ದೀರಿ. Okay, you can introduce, sir, and we will start the program, sir.
Yes, yes.
Okay, sir. Thank you, sir.
So, ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Good morning, everybody. So, this is second day of Science Week during 2023 PG Science Week. And today, for today's we have, we'll start with the morning session, plenary session. In the plenary session, we have our alumni, Dr. Murali Ramachandra. Of course, he was here as an alumni, our student during 79 to 85. So, Dr. Later, during those days, he was called as B.R. Muralidhara. Now, Dr. Murali Ramachandra is the CEO at Regain Oncology Limited, a wholly owned novel cancer drug discovery subsidiary of Dr. Reddy's Laboratory Limited. He received his PhD from University of Idaho, USA, and postdoctoral training from University of Kansas Medical Center and Dew Point Experimental Station. Prior to his role, he was the Chief Scientific Officer at Origin and positions of increasing responsibility of Shearing Flow Pharmaceuticals, USCA, and US National Cancer Institute. He has contributed to the identification of 19 innovative drug candidates that are undergoing global clinical development, one approved drug for the bladder cancer. This is one of the most significant achievement. Drug for the bladder cancer is, I think it's a contribution to the human kind. Murali is the author of over 70 research publications in the peer-reviewed international journals and inventor of 24 granted international patents. God, very good patent, sir. We'll appreciate you, sir. At least you are contributed to this human kind for their well-being. Of course, 24 granted great achievements. So garnering significant recognition with an overall citation about counting about exceeding 7,500 and H index of 29 and an I-10 index of 63. Currently serving as a member of a subcommittee on innovation, Indian Pharmaceutical Association, and Vision Group on Biotechnology and Karnataka. Dr. Murali, he was the alumni during 1979 to 85. During that time, he got University of Agriculture Sciences gold medal for the academic excellence during 1985. So he was a very bright student. Altogether, he's got the national fellowship for his higher studies. So on this occasion, sir, we welcome you, sir, to this program. Despite of your busy schedule, you have given us precious time, valuable time for us. So welcome to the GKVK campus after so many long years of your left the campus. Now we'll request Murali sir to deliver the lecture, sir. Welcome you, sir.
Thank you. Good morning to you all. First of all, thank you, Dr. Prakash. Thank you for inviting me, and it's a pleasure as a former student to address some of the younger students, because this brings me a lot of good memories from my days at UAS. So clearly, I think I'm very happy that I have this opportunity to speak to some of the postgraduate students today. So today what I would like to do is, if you give me a second, I'll display my slides. Do you see my slide?
Yes, sir.
So you're seeing slide number 2, right?
Yeah.
Yeah. So I think today what I would like to cover is 3 major topics. First of all, I would talk about my journey from US to cancer therapeutics company. And then I will talk about a little bit about drug discovery and development in India, specifically focused around cancer. And finally, I'll talk about, in that context, what we are doing at Origin Oncology, how we are trying to develop newer medicines to address some of the unmet medical needs of cancer patients. So you may all wonder, what's the connection between, I think maybe I'll close this window. So you may all wonder, what's the connection between agriculture, somebody who studied agriculture in his undergrad and then now working on a cancer therapeutics company. Here I've shown that I worked with Sharing Clove, which is now part of Merck Pharmaceuticals, one of the large pharmaceutical companies in the world, and currently working at Origin Oncology in Bangalore. So it may seem like completely unrelated, but if you closely take a look at it, you can see that if you look at how I eventually ended up in cancer therapeutics, after US, I studied, as Dr. Prakash mentioned, I did PhD from University of Idaho in the US. There I was mostly focusing on agriculture, microbiology, both US, Bangalore, as well for my MSc, and then at University of Idaho. Then I slowly moved into different disciplines in microbiology. I started studying virology, viruses, again, another discipline of microbiology. Interestingly, many of the viruses, especially DNA tumor viruses, they are the model organisms because they hijack the cellular machinery. They can induce tumors in certain host organisms. So because of that, I got interested in how tumor formation happens, what's the cancer, and then started looking at cancer biology subsequently. I worked at the National Cancer Institute, one of the premier cancer institutes in the US. And eventually that's how I ended up in cancer therapeutics. So in first few slides, I'll talk about my stints at different places, what I did and how the connection to a different discipline happened. So then you'll probably understand how from studying somebody who studied agriculture eventually ended up in a company focused on cancer therapeutics. So from 1979 to 1985, I was a student like many of you. You can see the picture on the left. So this is in front of the library building. I was also thinking I didn't know exactly what I was going to do, but I really wanted to do research when I do my MSc and PhD. That's something that I knew, but I was not sure which particular discipline that I would eventually end up with. So at US, after my BSc in agriculture, those days, Agriculture University, the main campus was in Hebbal. So this is the building many of you probably recognize in Hebbal. So this is where our third year and fourth year undergrad degrees used to be held. And then for MSc, I came back to GKVK. That's where I did MSc microbiology. MSc, I mostly focused on plant bacterial interaction, Azotobacter and Azospirillum interaction with plant, especially as you know that if there are some microbiology students, you know that these are nitrogen fixing bacteria. So that's what I studied at US. So from there, I moved on to University of Idaho for my PhD. So I still continued in bacteriology, but I started taking biochemistry as the second major. So there I looked at enzymology of lignocellulose bioconversion by soil bacteria. So I was still in sort of plant microbiology. I've shown one of the publications that came out of my work at University of Idaho. Interestingly, those days also, I had 2 international patents that came out of my PhD work. And one of them, here I've shown a newspaper article showing that this may have application in paper industry. This is a non-chemical way of bleaching paper and also degrading some of the paper byproducts. So this is an article in the local newspaper at University at Idaho. This was in 1985 to 89. So from there, I moved on to DuPont Experimental Station. DuPont, as you may know, it's one of the major chemistry companies, probably the biggest chemist companies at that time, chemistry company. They discovered a lot of polymers, including nylon and many of those things, Teflon, all these were non-stick vessels that, it's coated with Teflon. All these things were discovered at DuPont. When I was at DuPont, there were probably three or four Nobel Prize winners were there at the experimental station. There also, I focused on microbiology. I focused on bacterial conversion, degradation of xenobiotic. Xenobiotic are substances that are foreign to human or animal life, how they are converted by different bacteria. So I identified an enzyme called ferredoxin reductase that contributed to conversion of xenobiotic using a bacterial strain called Streptomyces graecosus. So I was still in bacteria, but specializing, also looking at enzymology, biochemistry of microbial conversion. So from there, I moved on to University of Kansas Medical Center as a postdoctoral research associate. At that time, I started getting more and more interested in maybe I should look at microbiology in the context of medicine, medical microbiology. So I selected adenovirus DNA replication as the research topic for my work at University of Kansas Medical Center. Adenoviruses are generally not so, they are harmless. They cause cold. These are viruses. These are DNA viruses. But interestingly, they are extensively studied to understand DNA replication because they do the replication within the human cells. So it gives a very good model system to understand how DNA is replicated. And there, in addition to adenovirus DNA replication, I looked at studied cell cycle control. This is the human cell cycle control, cancer biology, and this was one of the representative publication that came out of my work at University of Kansas Medical Center. And this is how typical adenovirus looks like under an electron microscope. So interestingly, adenovirus, in addition to while it is causing common cold, it also hijacks cellular machinery, especially the cell cycle machinery of cells. You know that cells have tumor suppressor genes. These are the genes or the proteins encoded by genes. They act as a strong brake. They prevent cancer formation in a normal, healthy individual. So adenovirus, it expresses certain protein. For example, one of the early protein called E1A made by this virus, it binds to one of the well-known tumor suppressor protein called retinoblastoma, the RB protein. The other protein called E2, or the E1B, that also binds to a second tumor suppressor protein called P53. As a result, it hijacks the cellular cell cycle machinery, induces cell cycle progression. Even when cells are not supposed to replicate, it induces cell cycle replication, and then the virus can replicate very easily because there are all the essential components required for virus replication is present if the cells are actively dividing. So essentially uses the cellular machinery, hijacking it for its own advantage to replicate and eventually produce a number of other offspring viruses to continue its infection cycle. So as a result, while I was studying many of these tumor suppressor genes and then critical transcription factors such as E2F, this activates number of pro-oncogenic signaling in cancer. These were all discovered around the same time while I was studying adenovirus DNA replication by this particular virus. As a result, these were all identified as the protein that interacted with the adenoviral protein. So that's how I started getting more and more interested in what's the biology in cancer cells, why in cancer cells. As you know, cancer is essentially uncontrolled growth of cells. When you lose control, cells start dividing and then growing excessively. That's what results in cancer. And then they start spreading. When they spread throughout the body, that's where I think patients succumb to the disease. So essentially, viruses also mimic to some extent for their advantage, although they may not cause cancer in humans. This particular virus does not cause cancer in humans. But still, if you look at the mechanistically or mechanism of action of how it hijacks the cell, it essentially works with many of the cellular machinery. That's how I started looking at various tumor suppressor genes, oncogenes, cell cycle control. I thought maybe cancer biology is something that I should start doing some more research and understanding more and more. That's where I decided to, and this is what happens when viruses hijack the cellular machinery cells. There is an uncontrolled cell growth, as you see in cancer. So from there, from University of Kansas Medical Center, with that background, I decided to go to National Cancer Institute, which is the part of National Institutes of Health. This is one of the premier institutions in the US. It's based in Bethesda, Maryland, where I extensively studied cancer biology and resistance to various approved therapeutic drugs. And what I studied there was one of the protein called human multi-drug resistance protein. This is a large membrane protein that pumps out many of the therapeutic drugs. As a result, therapeutic drugs is no longer active in cancer patients. So I started studying how this particular protein really functions in cancer patients. To study that, I wanted to have a system where I can express these proteins in any cancer cell lines in vitro, in cell culture, in the lab, to be able to express this protein and characterize this protein. That's where I started looking at, can I use some of the background that I had? Can I use the viruses as a vector to express the protein? So I made replication defective viruses. In this case, I used a vaccinia virus. I used a replication, and also I used two different virus system at that time. Other one is the baculovirus. These are the insect virus. In these cases, I put the P glycoprotein gene. MDR1 is a gene encoding this P glycoprotein. And then I could express using these viruses, I could infect any cancer cell line. I can see, now I can study what this particular protein does. So essentially now I combined my extensive background in viral molecular biology and use that to study the protein that are playing a critical role in cancer progression and conferring therapeutic resistance to many of the approved drugs. And this resulted in a number of publications. I've shown a couple of publications here. So that's how my journey, starting from Agriculture University, ended up in medical microbiology or looking at cancer biology. So from there, I went to a pharmaceutical company. I thought my major interest is in applied science, where I can really apply some of the scientific knowledge to come up with new and novel therapeutics. So I went to Sharing Flow, which is now part of pharmaceutical companies, which is one of the large pharma companies in the world. It may be number 3 or number 4 in the world as of now. Again, I continued my work on cancer therapeutics and gene therapy. So as I mentioned, adenoviruses hijacks the cellular machinery in order to replicate by itself. So what I thought was, together with my group, we worked on re-engineering adenovirus. Essentially, we expressed a artificial repressor so that in normal cells, it suppresses the replication of the virus. Once it replicates, it kills the cells. But what we made sure was in tumor cells, when you have a defective tumor suppressor pathway, only in tumors, it allows replication of this virus. Essentially, we came up with a replicating adenovirus as a therapeutics. And only in tumor cells, it replicates, it results in the killing of cancer cells. So essentially, all the understanding of adenoviral biology that I had from University of Kansas Medical Center experience and the cancer biology, again, I could combine all of these, come up with a oncolytic adenovirus to be able to use in cancer therapy. So this resulted in a very nice publication. We published that in Nature Biotech. It came as a cover article. In fact, the authors are, in fact, they had an expert write a commentary on this particular article, and he commented that this is an elegant example of combining research into basic aspects of cell cycle control, virology, and oncology. Oncology, as you know, is study of cancer biology and cancer therapeutics. So essentially, I think my group was able to demonstrate that you can bring in experts or knowledge from different disciplines and come up with a truly first-in-class therapeutics. And this, at that time, unfortunately, Sharing Flow decided to change the strategy, and this program went on the shelf. Now there are similar molecules that are going through clinical development. Parallelly, we also worked on a different approach. In this case, since I had worked on adenovirus, we can also make a replication defective adenovirus. You take some of the critical genes of the adenovirus, so then it cannot really replicate in human cells. So instead of viral genes, we put in a human gene called interferon alpha 2b. So this is a cytokine. Now with this replication defective adenovirus, we can use this as a gene therapy vector or the vehicle for gene therapy. So this was actually studied for bladder cancer. So essentially you deliver adenovirus carrying this human gene as a gene therapy for cancer. So I left around 2005. After this, this molecule was licensed out to a small biotech company called Ferring. And they eventually did the clinical trials with this vector. And just earlier this year, this got approved. This is the first gene therapy approved for bladder cancer. So I think I was happy that some of the work that I was involved from my experience or my time in the US eventually resulted in one of the approved product. So this is currently being prescribed for patients who have bladder cancers that are not refractive to, that are not responding to any of the available therapies or the previously approved therapies. So from there, I thought I always wanted to come back to India, do drug discovery in India. And with the background, with the experience, around the same time, there was also a lot of interest from one of the companies in India. And they were trying to convince me that it's better that if I can come to India, they can provide me, they can give me the infrastructure and all the whatever that's needed. So that's where I started looking at, can we do drug discovery in India instead of doing it in the US? Should I seriously looking at coming back to India and do the same thing in India? So I think with that background, I'll give you a little bit of a background about drug discovery and development in India. So India, as you may know, that it's considered as the pharmacy of the world. That's because we are the largest provider of generic medicine. When you say generic, these are copycat, or once the drugs are out of patent, patent is something that's the license to sell exclusively by your own, if you have a patent, we can sell a drug exclusively for 20 years since we apply for a patent. Then anybody can make a drug, then it becomes a generic medicine. India is the largest provider of generic medicines to the world, and we have about 20% share of global pharmaceutical export. But unfortunately, if you look at India, we are third globally in terms of the volume of drugs that we make in India, but we are 14th globally in terms of value. So I think that's unfortunate, because ideally we should be number within top 5, because we are making so much drug, but we are not really getting the value for the drugs that we make in India. That's because if you look at the evolution of pharmaceutical industry in India, we were mostly focused on generics or the copycat drugs for local market. It's also a good thing because you can make drugs much more affordable for patients. From there onwards, we focused on making large-scale drugs for other pharma companies. We were the global suppliers of pharmaceutical ingredients for many of the pharma companies. And then we started working on differentiated formulation, take a novel drug and come up with if it's an injectable, maybe try to make it oral, different formulation. We were also quite active in providing chemistry services to pharma companies of the world. But we were not really focused on innovative therapeutics in India. Only from 2005 onwards, there was a change in India became part of the World Trade Organization, or WTO patent regime. Till that time, India was not really recognizing a product patent. We could get a process patent, you can make a drug and get a patent. But if you discover something new in India before 2005, you could never get a patent in India. As a result, innovation was never really supported in India. It's only after 2005 when there was a change in thinking that India became part of WTO, then there was NCE, or the Novel Chemical Entity research, started happening. Now I think there is a lot of emphasis in terms of India can also be an innovation-driven global pharma company that will help in increasing the value of the products that we are making in India instead of simply focusing on generic medicine. So if you look at different companies in the world, so on the left, I've shown various companies from US and the Western world, 25 companies on the right. These are the ones that are focusing on generic medicine. If you look at top 25 companies, and based on the value, you don't see any Indian companies at all. But if you look at the companies that are focusing on generic medicine, many of the Indian companies, including Sun Pharma, Dr. Reddy's, where I'm currently working, these are all there because we focus on generic medicines. These are out of patent. Essentially, they are not giving as much value, but this is helpful for patients because you can make it much more affordable. But you can make a generic only after it goes out of patent. Innovative drugs, when it becomes out of patent, one can make a generic drug. Essentially, these are copycat drugs. So this highlights that we should also start looking at developing innovative therapeutics in India. because you know that India has a very young talent pool. We have a very strong chemistry foundation, and there are a lot of returning Indians like me, and there are many NRIs who are exposed to best practices in US and other countries. They want to replicate some of those successes in other countries in India as well. And if you look at in India as a whole, I think after we got independence, we had a green revolution. Many of the agricultural scientists, including I'm sure people from US, contributed to green revolution. We became self-sufficient in food production. Then over the years, I think we have seen in Bangalore being one of the centers of IT revolution that helped India grow dramatically, I think, in leaps and bounds. What I believe is now there is an opportunity for us to replicate some of the success we have had in IT industry, in pharma industry as well. And as I mentioned, now the intellectual property rights or the patenting that's similar to what happens in the US or in Europe. So that also should foster innovative drug development in India. And one thing that's unique, as you all know, that we can do discovery, our India operation is significantly more economical, it's more cost effective. If you were to succeed in drug discovery and development, once you have a successful drug, it doesn't matter where it comes from. I think it should treat patients, whether patient is in India or patient in the US. So we can leverage the economy of India operation, especially if you look at clinical trials. I'll talk about when the clinical trial happens during drug discovery development. As you know that India has large number of patients, we can do clinical trials in a cost-effective manner. And also there are very good clinical centers, especially if you talk about cancers, there are very good clinical centers such as All India Institute of Medical Institute, you have Tata Memorial Center in Mumbai, you have a lot of other very good cancer hospitals in India. So there are with very good scientific capabilities and infrastructure. So idea was, can we leverage some of the unique advantages that we have and start discovering innovative therapeutics in India as well? So in addition, I think there are two major reasons that India never really succeeded. I think the patent was one of them, but the other thing is lack of funding. If you look at the cost of drug development for a successful drug, modern drug, it takes close to $2 billion, about $1.7 billion. And if you start with about 1,000 compounds, one of them will end up in the clinical development, and only 1 out of 10 that enter clinic will eventually succeed. So there is a lot of failure in drug discovery and development. And also you need 10 to 15 years to successfully develop one drug. So this is one of the drawbacks. We can't really afford to spend this kind of money being in India. If you look at total R&D budget in the US, for example, for pharma is about 50 to 60 billion. China is about 15 to 20. In India, we are spending only about 3 billion. So with this, it becomes extremely challenging to do modern drug discovery and development in India. And also we don't have the expertise as much as in US or Europe because we were never really involved in innovative drug discovery. So if you look at specifically what's happening in cancer, most of the early cancer treatments were surgery. You just take out the cancer, chemotherapy, using toxic chemicals, you sort of ablate cancer. In the recent time, it's targeted therapy, whether activating somebody's own immune system to take care of cancer, or you go after specific mutations or specific changes that are seen in cancer. Initially, you also know that radiation therapy, these were all there. These were all sort of a brute force approach, but only after 1990 onwards, I would say there is a lot more focus on targeted therapy, where specifically and looking at the mechanism in cancer, people are trying to address, can we come up with novel therapeutics? So I think here I've shown the difference between traditional and modern drug discovery. In a traditional drug discovery, you would simply look at many cases at serendipity from you see that any specific compound or drug, does it show desired activity in an animal or a cellular model? If so, then you sort of develop it as a drug. Whereas in modern drug discovery, with a advanced understanding of basic science and the disease biology, there is a clear paradigm shift in drug discovery nowadays. We know that what are the proteins that are altered in cancer setup? Can we, in vitro or in the lab, can we identify drugs that are modulate this particular protein that are dysregulated in cancer? And can we develop this as the drug? So this is the emphasis in last 15 years or so. So typically in a drug discovery, to start with, we look at, let's say in this case, we focus on cancer. we try to understand the molecular mechanism of a specific type of cancer. And then we look at, are there any specific protein that we should go after? It could be an enzyme, it could be gene itself, it could be a receptor on an ion channel, nuclear receptor that's dysregulated in a particular type of cancer. So once we identify this kind of something that's altered in cancer, then we need to validate it with a knockout animal studies or various approaches. We make sure that if you were to modulate that particular protein, you get the therapeutic benefit. I think if you can demonstrate by whether genetic means or other systems, then we start with the drug discovery using this particular target. It could be an enzyme or a receptor. So if you look at cancer, there are a number of things that are dysregulated in cancer. It could be uncontrolled growth. It could be avoiding immune response. It could be having immortality. It could be inflammation that's contributing to tumor growth. It could be metastasis or spreading of the cancer cells to different organs. A lot of changes. So if you look at specific hallmarks of cancer, there are very well-known targets. Again, these are emerging areas. There are newer and newer targets are identified. So these are some of the examples of the proteins that are considered as good targets for cancer drug discovery. So once you identify a target, if it's an extracellular, it's outside of the cell. If it's a soluble protein, you can come up with an antibody or a peptide or a small molecule to modulate that particular protein. If it's a membrane bound, then you ideally go with an antibody or a peptide or small molecule. If it's inside the cell, then you can't use antibodies. You have to use small molecules that can go inside the cell and modulate that particular target, or you can use antisense, RNA, interference, some of the other approaches. So typically, I think depending on the location of the target, we decide on whether it's ideally suited for a small molecule or an antibody approach. So we can control that particular target by different means. If it's an enzyme, we can inhibit. In some cases, you may want to activate it. Again, small molecules or antibodies or peptides can do it. And some of the newer technologies where we can simply degrade the target of interest. Let's say there is a particular enzyme that's dysregulated in cancer. We can use a small molecule to completely eliminate it. And we can suppress the expression of protein by antisense and or RNA interference methods. We can modulate DNA, mRNA, we can overexpress. So this is what happens in gene therapy. For example, the gene therapy product that I mentioned, where we express a certain particular cytokine that gives you anti-proliferative activity. It's a DNA or a gene therapy. And antibodies are some of the most popular these days. With antibody, they can bind to the target of interest, because you know that antibodies can bind to any protein of interest. And there are different ways of killing cancer cells using antibodies. So typically what happens in a drug discovery is once we have a target, we need to make the protein in large quantities. We use a different expression system. It could be making it in bacteria, insect cells, or mammalian cells. We purify them. Once we have the purified protein, it also helps us in identifying the 3D, three-dimensional structure of this protein. Using that, we design the compound, molecular modeling informatics people, and they'll design the compounds. Chemists make those series of compounds. Then we start looking at, does this compound really modulate the protein or the target of interest in a biochemical or in a cell, in a test tube? So that's in biochemistry. Various assays are done. Once we identify compounds that are modulating the target in a test tube, then we start asking, does it work in a complete cellular system? Does it go inside the cell? Does it really modulate the target in a cancer cell? And that's done in a cell and molecular biology group. So once we have a drug, a molecule, small molecule compound, for example, doing all these things, then we start asking, does it have drug-like properties? Because you want to make sure that in many cases, you want an oral drug, you want to make sure that it's orally absorbed, it has the right pharmacokinetics. Pharmacokinetics is how long drug persists in a human body, what's the elimination route, does it stay in the body for too long, what kind of safety data. So these are all studied in drug metabolism and pharmacokinetics group. From there, we start looking at, now we know that it has the right drug-like properties. If you inject that into an animal model, it has the right exposure, it gives you certain hours in a day so that it can engage the target and give the therapeutic benefit. Then we start looking at the disease model. We have tumor models established in animals where we start looking at the efficacy, therapeutic benefits of these compounds. And then parallelly, we look at what kind of side effects, because you want the drug to be efficacious, killing cancer cells without impacting normal cells. It should have a very good tolerability. Toxicology is also done in animal models to start with. Then we develop the process to make these drugs in large quantities, in kilogram and in much bigger quantities. Parallelly, we make sure that we can patent these molecules. We get complete intellectual property rights for these. So we need to have those capabilities as well. And once we make sure that we have a drug that's safe and efficacious, then we take it to human clinical trials. Human clinical trials are probably the most expensive part of drug development. And you need 3 phases. So if I can expand on human clinical trials, in phase 1, we simply look at in a smaller number of patients, what's the maximum dose we can administer with very good safety profile. Typically, this is less than 100 patients. Then we go to phase 2. This is typically several hundred patients. Again, in this case, we start asking, does the dose we identified give you therapeutic benefit? In this case, in our case, does it give you anti-cancer activity? And then we have to show in a large number of patients, many times it could be thousands of patients, where we need to demonstrate that it is indeed active to be able to get approval from either Indian regulatory agencies or US FDA. So this is the most expensive. Typically for these studies, each patient, we need to spend up to $50,000 to $100,000 per patient. That's what makes drug discovery and development extremely expensive. So I think with that background, Dr. Reddy's, some of you may know that Dr. Anji Reddy, he was the one who is a PhD in chemistry. He founded Dr. Reddy's lab. Similar to many of us who ended up coming to UAS, he was also born in a small village, but he had a very strong desire to set up a largest pharmaceutical company in India. And now it's, I think, one of the top 5 pharma companies in India. And his major desire was to make sure that we discover and launch a drug globally from Dr. Reddy's companies. Unfortunately, he passed away in 2013 without getting any drug approved, because as I highlighted the challenges in drug discovery and development, he was the first one in India to start innovative drug discovery in 2003 or so. But I think he had a very strong desire that we need to discover and develop innovative therapeutics in India. That's when I think some of the senior management people from Dr. Reddy's started speaking to me whether I can also join this particular vision of Dr. Reddy's, come back to India and start working on innovative therapeutics. With that background, I came back to India in 2005. To start with, I was the head of biology, then I became chief scientific officer. And since last 4 years, I'm the chief executive officer of Origin Oncology, which is the innovative cancer therapeutics subsidiary of Dr. Reddy's. So Dr. Reddy's is a leader in generic medicine, but our subsidiary focuses only on innovative therapeutics. I think the track record that we are extremely proud of is we have contributed to about 19 innovative therapeutics that are undergoing global clinical trials. From Origin itself, I have more than 35 publications, 17 granted global patents, and a couple of publications I have listed here. One is in Nature, other one is Nature Communication Biology. These are just representative publications. We've also been covered in some of the Indian newspaper. For example, this was an article earlier this year from in a magazine called Shastra. This is published by IIT Madras. So they covered some of the innovative therapeutics work that's happening at Origin. So I think just to give you some more background about Origin Oncology. So we've been around since 2002. I joined in 2005. Our expertise is in small molecule antibodies and cell and gene therapy. We have a track record of more than 80 discovery programs. Currently in India, we are the leader in innovative therapeutics. We have more than 200 new chemical entity global patents. We have 19 compounds that are in clinical development, and we have a very strong pipeline of clinical as well as preclinical. When I say preclinical, where we are characterizing these molecules in animal models. So essentially, to address the low probability of success and the high investment needed, what we are doing is we are generating a very strong preclinical pipeline. Some of these molecules we partner with global companies. As a result, we get significant upfront and we get R&D funding and milestone. And with the cash flow that we generate, idea is we will keep some of the molecules and develop it all the way to commercialization, global launch eventually. As a result, part of our assets are partnered. And When we partner also, we keep India rights and rights for some of the other countries, and we have royalties and other milestones when we do this kind of partnership. So these are typically high value partnership that brings us cash flow to be able to put together a very strong pipeline of therapeutics. So we work on 3 different therapeutic modalities. As I mentioned, small molecules is one of the expertise. We have also started working on antibody-based therapeutics as well as cell and gene therapy. So in small molecule, I think there are some unique platforms that we have set up at Origin. For example, this is the small molecule oral immune checkpoint platform. If you look at some of the most successful drugs in cancer in the recent years is the antibodies that are targeting protein called immune checkpoint proteins. Essentially, by inhibiting this particular protein on cancer cells, one can activate patient's immune system, which can take care of cancer. But the problem is this antibody approaches, these are injectable. You have to make it in a recombinant way. As a result, it becomes extremely expensive. Typically, it's about 150,000 per patient per year. So what we decided, maybe we can come up with a new way of making this in a small molecule approach. These are smaller molecules. We can chemically synthesize it. As a result, cost of goods is significantly lower. We can make these therapies much more affordable for patients. And this could be oral because they are smaller molecules. We can administer them as a capsule or a tablet. And we have demonstrated this platform, and we have a couple of molecules that are in clinical development as of now. One is in phase 3 advanced clinical trial. So the other unique platform that we built was, I mentioned that there are newer approaches where we can simply, inside the cell, we can go and eliminate the dysregulated protein. So essentially, we can make it disappear. This is by using a bifunctional molecule. One end of the molecule binds the target protein, other end targets this protein to a proteasomal. This is the normal cellular protein disposable machinery. As a result, when you have this bifunctional molecule, it degrades the target protein. Some of the unique approaches that we have brought into this approach is we have come up with a structure-based approach where we can come up with this bifunctional molecule without any linker. As a result, these are extremely small. They are much more amenable for optimization. In fact, we have published one of our molecules last year in Nature. They were surprised that you can come up with a small molecule that's capable of degrading the target with such a small size. I think this is one of the unique platform that we built at Origin. Another approach that we are taking is the antibody-based cancer therapeutics. So with antibody, you can simply bind to the target protein on the cell surface, and then you have other immune cells coming and eliminate these cancer cells. We can make a bispecific, one arm of the antibody binding to protein on cancer cells, other end binding to the immune cells such as T and NK cells. As a result, cancer cells are killed. There are other approaches where we take an antibody, conjugate with a cytotoxic drug. It can deliver this cytotoxic agent to cancer. This is much more specific compared to standard chemotherapeutics, where you don't have the targeted delivery. So this is another approach that we are doing. We are also looking at engineered T cells that I'll talk about in the next slide. In this case, we identify an antibody that can bind to a target in cancer cells. We take only the smallest fragment that has the binding region. We make a chimeric antigen receptor. We genetically engineer T cells. So essentially what happens is in this kind of therapy, chimeric antigen receptor or the CAR T therapy, we isolate T cells from patients, and we genetically engineer in vitro in the lab, and we expand them and we put them back into patients. This is highly individualized therapy. And now these T cells, because they have this chimeric antigen receptor, they can recognize only cancer cells and kill cancer in this particular patient. One of our CAR T trial is going to start within the next couple of weeks. We have the approval already. Our trials is going to start in a couple of centers in Bangalore to start with. So I think these are the therapeutic modalities, therapeutic approaches that we are taking. If you just quickly walk through what we have done over the years. So 2005, we had a significant management change. That's when I came in, and we brought in a number of collaborators to start with. We worked with some of the big names in pharma, including Merck, Novartis, Johnson, AbbVie. What it helped was, as I was telling you, that we don't have expertise in India. So a few of us came back with the drug discovery background in the US. And with this kind of collaboration model, it helped us in strengthening our organizational expertise in training large number of people at Origin to do drug discovery and development. It also helped us to establish a profitable company and without having to depend on other investors. So these are the companies that we have worked in. You can see that at least 6 out of the top 10 pharma companies that we have worked with. As a result, we have very good recognition, very good visibility in the pharma field all over the world because they recognize Origin because of our extensive collaboration with these companies. So these are some of the programs our partners, Novartis, Orion, or the other companies shown here. They are developing, they are in different stages of clinical development. In these cases, we only did the discovery. We transferred the patent rights to these companies and they are doing the clinical development. So from 2009 onwards, what we said was, if we have the capability, maybe we should develop on our own. So we built a pipeline where we started the program on our own. Some of the program we partnered with companies shown here. And now we have a very strong proprietary pipeline of novel assets. Initially, we focused on cancer as well as inflammatory diseases, but currently our focus is predominantly in oncology or the cancer therapy. So these are some of the assets that we licensed out. This helped us to bring in even much higher value to the company, higher cash flow to the company to build even a stronger pipeline. So again, I don't think there is any other company that has licensed out this many assets to other companies. Here also probably we have a unique track record. And one of the assets that we licensed out, this is a small molecule inhibitor that inhibits a kinase called IRAK4. This was looked at in AML by our partner, Curis. It's a Boston-based company. So in the initial stages itself, about 12 patients were dosed with this drug. All of them, irrespective of the dose that was administered, showed a reduction in the cancer cells. This was quite dramatic. And I think this was recognized by all the experts in the cancer biology, as well as even the stock market in the US reacted to this kind of positive news. The market cap, the value of this company within 2 weeks of time, it went up from 60 million to 1.3 billion or so. So this is what a good drug can really make a difference to a company. So they are developing this drug, but we have rights for India, Russia, and few other geography. We have royalties once it eventually gets approved. This is just to showcase that some of our drugs that we have licensed out are doing very well in the clinic. So here I've shown all the other drugs that we are doing. They are in different stages of clinical development, phase 1, phase 2, phase 3. The ones shown here on the top, these are all clinical trials that we are doing on our own. Ones in the bottom, these are clinical trials with our partners. And here are some of the programs, some of our clinical data that we have generated. This is one of the drug. This is probably the only program where we are looking at inflammatory diseases. In this case, we are looking at severe psoriasis. Psoriasis, as we may know, it's a severe autoimmune skin disease. There is no cure for that. And the approved drugs are the most effective drugs are antibodies. Those are injectable. We are trying to come up with a small molecule drug that can be taken by oral route. Here you can see that in our clinical trial, phase 2 trial, 2 patients in about 12 weeks of dosing, psoriasis lesions are completely gone. So this is quite encouraging, but we have to demonstrate this in a larger number of patients to be able to get the approval. Here is another example. This is a kinase inhibitor. It inhibits some of the key oncogenic kinases. So in our phase 1 study, you can see that these are the number of patients, and the ones that are shown below this line, these are the patients where tumors are shrinking because of the drug. This is again an oral pill. And interestingly, we have recently found that I was mentioning earlier that immunotherapeutic drug is one of the successful drugs, but in some of the cancers, they don't really give you maximum benefit. For example, in this tumor model, the immunotherapy drug is about 50% active. Our drug is also 50% active. If you were to combine in this model, most of these animals became about 40% completely tumor-free. This is quite remarkable. Potentially, we can combine this with some of the best-selling cancer drugs and improve the efficacy and durability of response in cancer patients. Parallelly, we also looked at in fibrosis. As you may know that even in COVID infection, fibrosis, the lung fibrosis is major side effect. And unfortunately, we did not have this drug at this time. This is an animal model for fibrosis, lung fibrosis. this molecule shows very good efficacy. There are no good approved drugs for this fibrosis indication. We are now developing this particular drug for fibrosis as well. So this is another drug. This is in lymphoma and lung cancer. You can see that when patients are taking this drug, they are surviving longer without any. These are patients who have exhausted all other therapies. If you just don't do anything, in about 8 weeks, most patients die at this stage. But now with our drug, they are surviving longer. What we are doing now is combining our drug with one of the chemotherapy and looking at in lung cancer. And this is in a CT scan, you can see that tumor is completely gone by taking this oral drug. This is very well tolerated, no safety issues. And we are developing this drug. We are doing phase 3 trial with this particular drug. And we have a number of other programs that are in preclinical stage where we are looking at evaluating the efficacy and tolerability in animal models. Again, total we have about 30 plus programs at Origin. So I think finally, I think people are recognizing some of the innovative drug discovery and development that are happening at Origin. This was, I think, an article that got published last week in Economic Times. They covered some of our CAR-T or the cell and gene therapy efforts. I think they have also highlighted one of my quotes. We are trying to make some of these therapies much more affordable so that patients in India and other emerging economies also benefit from these novel therapies. And I think with that, I'll conclude. Hopefully, I have conveyed that scientific disciplines are highly interconnected. Many of you may be thinking of doing something a little different, but I think once you understand, once you start building expertise in a scientific discipline, it's much easier to make switch to something else that you are much more passionate about. I think probably my example is a case study in that particular aspect. And I think as a result, career options are limitless. I think if you do well, if you were to focus on what you're doing, I think your career options are limitless. But you have to have whether it's a typo, you have to have an open mind and see where you can see an opportunity. And also, second point, this is probably there is a misinformation. I think a lot of people think that you can't really do good research in industry. I think probably I have conveyed to you that you can also do good research in industry, not only come up with impactful publications, but also more importantly, come up with highly impactful therapeutics that can address the unmet medical needs of patients, not just India, worldwide. I think that kind of profession, even in industry, could be highly rewarding. So again, I think some of you are much more passionate about applied research. You should not shy away joining industry because we can do good research in industry as well. I think with that, maybe just last slide, even in drug discovery and development, there are various expertise that we depend on. For example, there may be people even at UIS who may be experts in biochemistry, entomology, and some of the other aspects. We have a lot of people, for example, veterinary graduates from my time as well, BVSC graduates at Origin looking at pharmacology, how a drug behaves in an animal model, to looking at toxicology. So there are a lot of expertise that one can utilize in drug discovery in a pharmaceutical company. I think with that, I'll end, and then I'll be happy to answer any question that you may have.
Thank you, sir. Thank you so much, Dr. Murali. Of course, it's an eye-opening talk. What we do not know about the drugs and your innovations, really a good thing for all of our students. Of course, the health depends on the food what we take. The food what we are taking today is really, it's very chemical based, everywhere pesticide, herbicides. Again, it's a more alarming. Cancer patients are increasing. In that direction, you have come out to India. India is an alarming trend for cancer patients. That you are trying to give very affordable, cheap prices drugs that you have mentioned. Many cheap therapies also you are giving. So on this occasion, it's a very good contribution, sir. And I think I will.
Few things are there.
Yes,
sir. Sir, I'll just take over at this moment. Sorry for disturbing you. Sir, it's really exciting to know and follow your journey, which has started from USB, sir. Really, it's mind-blowing and eye-opening, and wherever that we can go. And as you are as you have given that the industry is the place wherein we can be able to think about for your future and other things. Definitely our students also looking for the same kind of opening, and they are also moving towards that. And really it's scintillating to know about the different avenues that what you are exploring for the management of cancer. Really it's very amazing, sir. So there are a few question and answers which they have posted. I will just read out on their behalf, sir. The first one is that from Professor Tambad, who has got a query saying that that immunotherapies is effective in cancer treatment compared to chemotherapy. What is the recovery rate in the immunotreatment? That what he has questioned, sir.
Even in immunotherapy, not all cancers respond very well.
Yes.
In most of the cancers, even the best response, only about 15 to 20% of the patients respond. But if you respond, the efficacy of the response is durable. But in some cancer, for example, in Hodgkin's lymphoma, there are some unique cancers where they are highly effective. You get about 80, 90% response rate. But right now, the challenge is how can we improve the response rate of immunotherapy? That's where we are looking at various combinations. By combining, can we increase this 15, 20% response to say maybe 90 or even 100%? So that's where, including CAR T therapy, that, for example, I talked about, there the response rate is 80, 90%. That's also a sort of immunotherapy. And again, but CAR T therapy, I did not comment. Globally in the US, it costs up to $1 million per patient. And we are trying to make it much more affordable. To start with, it could be anywhere some 50 lakh. It's still very expensive for patient. But our goal is to bring it further down by about 75% further. Then it become much more affordable. But clearly, I think there are a lot of good activities. Different combinations probably will address the response, lack of response in some other patients.
Yes. So that's what the other question was from the Professor CNL Reddy. He has asked the same things, a CAR T therapy with the rest of the therapeutic options. And what is the cost that what you have explained right now, sir? So other thing that drug resistant is most major problem in cancer therapy. Will the companies keep it in the scheme while developing the new drugs? Is there any way the cancer drug can be combined with other molecules which can delay the resistance development or reversing the resistance development?
Yes, that's clearly one of the focus in cancer therapeutics and innovative cancer therapeutics. Because they grow so fast, there is so many mutations that are happening. Resistance is almost universal. It depends on how long it takes before you start seeing resistance. But I think with some of the immunotherapy and other approaches, direct tumor targeting approaches, I think it's possible to delay the occurrence of resistance. Again, that's one of the key focus of approach for us as well. How can we delay the resistance occurring in patients? And I think that's where I think combination is probably the way to go.
Students and professors from plant biotechnology want to know whether these nanoparticles been used in the cancer therapy.
Certainly, for example, with many of the approaches, this is just one example. For example, with nanoparticle, you can make a targeted delivery to tumor. Maybe there may be certain agents that could be effective, but it may not discriminate between tumors versus normal cells. You can use nanoparticle to specifically target to tumor cells. In other cases, maybe certain drugs, ideally you may want it as an oral drug, but they may have very poor absorption through the gut. In that case, maybe various nanoparticle approaches might give you the oral bioavailability. So nanoparticles do have a role to play in innovative therapeutics as well.
Yes, sir. And finally, there was one more question which all of your slides have given that one. In spite of that, one of the faculty has given. Is there any research work on gene therapy with genome editing of cancer genes?
Certainly. So with gene therapy, the challenge is, in fact, when I was chairing flow, I mostly focus on gene therapy. The main challenge is, if it's in a confined region cancer, then it's possible to go and introduce a gene of interest or do the gene editing. The major challenge is delivery. Can we have a vector or a system where we can deliver the gene editing tool or the therapeutic gene to all the cancer cells? That's where the challenge is. For example, the bladder cancer that I talked about, it's a cancer within the bladder in a confined area. So there, by interferon gene, it's actually a secreted protein. It comes out even if few cells are where we have the gene therapy, the protein is made, it comes to the extracellular region, and as a result, even other cancer cells are impacted. So I think there are niche areas where it could be applicable, but still we need to address the delivery issues. That's where even nanoparticle, as you know, that with mRNA vaccine, the delivery, what really made it successful. Nanoparticles were over there as well.
Sir, I have Professor Nataraj Karba with me. He is having some question. He will post that.
Sure.
Dr. Murali, excellent talk. Thank you so much for accepting our invitation. My query is that you mentioned in one of your early research that this adenovirus hijacks basically the cell cycle machinery. And what happens to the checkpoints? You have now G1 to S phase checkpoint there. So do you think those things are suppressed totally? Therefore, they hijack the machinery?
It's completely suppressed because, as you know, that because of P53 tumor suppressor gene, that's a checkpoint protein. If there is damage to the cell, it arrests cells and gives enough time for the cell to fix it. But what happens if you bring in the virus? Virus makes a protein called E1B that binds to P53, makes it completely inactive. You lose the checkpoint in infected cell.
Yeah,
So
great.
that's how, in fact, P53, a lot of early functions were characterized based on its interaction with the viral protein. That's what was so fascinating to me to start looking at cancer biology as a potential career option.
Great. Could you attempt to target this kind of machinery during our cancer cell division? Like
That's
targeting
the
the
oncolytic
checkpoints?
virus that we published in Nature Biotechnology. That's one of the attempts to do that. Essentially, what we did was, because in cancer cells, P53 is mutated, it's inactivated. So we made a virus, wherever P53 is mutated, it replicates uncontrollably. When there is wild type P53, it is completely held in check. That's the virus that we created. That's what the title of the article was, Re-engineering adenovirus pathway for a tumor-specific killing. So I think the same idea that we adopted in that particular engineered virus.
Oh, sorry, I missed the paper. Thank you so much. I hope I get a chance to interact with you more. Thank you. Back to you.
Sir, thank you very much for your nice and very good presentation that what you have done. So far, 156 members they have joined to witness this particular talk, and finally remained on 112. It's very, very scintillating and it's very heartwarming to note. And on behalf of the university,
Prakash, 1 minute.
yes,
I have one more question for.
Yeah, please, sir, please.
Sir, usually the patients who are suffering for the hypertension and the sugar, but diabetic, they are taking much of medicines. So such medicines causes for these the cancer.
No, unfortunately, I think there is no, because those are all fairly extensively tested. None of the, or at least at the therapeutic doses, they are not known to cause cancer, because those are all long-term studies are conducted and then only it got approved. In fact, for example, metformin, which is one of the diabetic drug, it has anti-cancer properties. But many of them, I think that's where phase 3, these are large trials, thousands of patients, long duration trial, especially if it's a metabolic disease such as hypertension, diabetes, it has to be multi-year trial. And finally, if it has the approval from US FDA or Indian regulatory agencies, and you can assume that it's relatively safe to take these drugs.
As a patient, myself as a patient with these two aspects, why is much of burning is there, acidity is there. Due to acidity, is it not causes anything like the tumor, something like that inside the gut?
If you have chronic acidity, it may lead to some, but again, I think you have to make sure that there is where chronic acidity is not there. But there is no direct connection as such. But anything, if you have constant insult, constant damage to cells, it has a potential to turn into cancerous. So that's where you should not have those kind of things that are there constantly. I think once in a while, acidity and all that, it's okay. But you have to control that as well, I would agree.
So, one more question.
Yes, I'm Dr. Satish from Indian Institute of Horticulture Research. Presently, there are a lot of technologies which have come up with the gene silencing. Is there any work on that gene silencing, and have a product like that? Because certain particular level of traits can be silenced through your genes.
Yeah, but not in, again, the challenge is the delivery. How can you silence whether it's siRNA, shRNA to a large number of cells to get the therapeutic benefit? That's where I think delivery systems need to be improved. In a smaller indication, for example, siRNA, shRNA silencing approaches in some ocular diseases where it's in a confined space, they are approved. But in cancer, to be successful, you need to make sure that delivery is improved. They can be delivered to relatively large number of cells. So that's the challenge.
Okay. The another issue is like some of the even antacids established like ranitidine. It has got some contaminants like nitrosomonas compounds, which may become carcinogenic and become causing cancer. Is it having awareness among physicians or chemists?
Yeah, so clearly, I think with the ranitidine, with that particular, it's actually an impurity that cancer causes.
Impurities.
Yeah, it also causes DNA damage. So
Yes.
that's something I think there has to be clearly awareness. I don't know how much awareness is there.
Yeah, but in India, as such, there's not much awareness, but probably American and European countries, there are, I think, almost it's a band, I suppose.
Yeah, I think that's where
in India, among physicians and chemists, I think now it's going on. Like, this is what just I would like to know.
I think people who are familiar, we should probably try to educate everybody around us as much as possible. In fact, I've told, as soon as I learned about everybody that I know, don't take this drug anymore. There is a small impurity that can potentially cause cancer and all of that. So
okay. Thank you very much, sir. Thank you. So my daughter is doing research on this ranitidine.
okay.
Yes. So thank you for the participants. If you have any queries, please mail back to us so that we will forward to the Professor Murali so that we can get the whatever the required answers and we will let you know. So this is what we would like to inform for the participants. Otherwise,
Murali sir.
Yeah.
Please visit physically to the campus, sir. We'll invite you so that you can address your microbiology and pathology students.
Sure, I look forward to that opportunity.
Yes.
I was there about 3 weeks ago.
Definitely we'll make the arrangements for that.
About 3 weeks ago, we had a 40-year celebration, but that was on a weekend. Sunday, I was there for a couple of hours. I was there in the auditorium. It was nice to sort of reminisce all the nostalgia that from US Bangalore. So.
Yes.
Thank you. Thank you.
Thank you very much on behalf of the directorate and on behalf of the university. We profusely thank for accepting this invitation and providing your insights, sir. And wholeheartedly, the university compliments for you for your whatever the significant achievement that you have made in your scientific journey. And we wish that you will also fly much more in colors with that and come out with some more products. So on behalf of the university, finally, I congratulate and compliment and thank you for the participation in this Science Week and igniting our student minds and providing this opportunity for every one of us to interact with you. Thank you very much, sir.
Thank you. Thank you for
Thank you, sir. Thank you so much.
Thank you for all the kind words. I really appreciate it.
Thank you, sir. Thank you very much.
Bye-bye.
Now it's open to everyone. Please, the oral and poster session, let us continue with that. Thank you very much, sir. Please.
ಈ ನೋಡಿ ಸರ್ ಈಗ ಅವನು ರೆಡ್ಡಿ ನೆಕ್ಸ್ಟ್ ಸಿಇಒ
ನಮ್ ಗ್ರಾಜುಯೇಟ್
ನಮ್ಗಿನ ಸೀನಿಯರ್ ಮುರಳೀಧರ್ ಅಂತ ಸರ್ ಮೈಕ್ರೋಬಯಾಲಜಿ ಸರ್ ಸರ್ ಅವರು ಹಂಗೆ ಅಲ್ವ ಸರ್ ಕೃಷ್ಣೇಗೌಡನು ಹಂಗೆ ಈಸ್ ಆಲ್ಸೋ ಫಾರ್ ದಿ ರಾಷ್ಟ್ರೀಯ
Dean PJ Saurabh. Hello.
Sir, really, sir.
What is the next program?
So next program is this next oral presentation and the poster presentation, sir, from 10:30 onwards.
Oral presentation online ಅಲ್ಲಿ ಇರ್ತದಲ್ಲ?
ಇಲ್ಲ ಸರ್, ಇಲ್ಲ. ಇರುತ್ತೆ ಸರ್, ಇರುತ್ತೆ. ಅದಕ್ಕೆ ಕೊಟ್ಟಿದ್ದೀವಿ.
ಓಕೆ.
ಇದೆ ಸರ್, ಇದೆ. ಇರುತ್ತೆ ಸರ್.
ಓಕೆ, ಚೆನ್ನಾಗಿತ್ತು ಇದು.
ಚೆನ್ನಾಗಿತ್ತು ಸರ್. ಮುರಳಿ ನಮ್ಗೆ 1 year ಸೀನಿಯರ್ ಸರ್ ಇವರು.
ಓಕೆ.
Thank you, sir.
Right. Thank you.
Thank you so much, sir. Thank you, sir.